Sting is a sensor. It senses DNA in tissues and fires off a signal which tells other cells what to do. Most people think that it tells the immune cells to activate so that they fire off and cause the immune system to respond. We found that the opposite occurs, that through the same molecule that other people have found activates the immune system, that same molecule, called Sting, will actually shut down the immune response. It has important implications for how we understand disease processes. In this particular case, how we understand where autoimmune diseases come from. So once we know that Sting is a target for turning off the immune system, that means that in principle at least, we can use reagents that we could call Sting activators to shut down the immune response artificially. It would be a good idea where the immune system is hyperactive, and that's these syndromes I'm referring to, autoimmune syndromes, which are familiar ones to most people, are type 1 diabetes, rheumatoid arthritis, multiple sclerosis, and many other syndromes are like that. So they're diseases which are caused by the immune system being overactive. So those are the particular settings where we want to turn it off or switch it down. The general treatments are that we globally turn off the immune system with drugs called immunosuppressants. The bad thing about that is that that's going to shut down all the good things that the immune system does at the same time, like fighting off infectious disease. So people who have these drugs given to them have more risk of having infections. Well, I think with Sting, and more research to come, because we still don't have all the answers in this, is to target this turning off the immune system to particular tissues at particular times, so that we can be more specific about how we control the immune response. Well, the further targeting we can do, since we're talking about autoimmune disease, is that in some of those diseases it's known what the target tissue is. For example, in type 1 diabetes, it's very small numbers of cells in a tissue called the pancreas, and those cells make the hormone insulin. And so we know that insulin is one of the targets of the immune system. So we can actually feed the immune system with this same thing, insulin, and ask it to not respond to it, but to turn off that response to it, so that the immune system is very specifically and in a very nuanced fashion turned off against that particular disease target. Sting is just part of the answer here. It's a component that we didn't know existed in this way before, particularly as the field had actually shown in several systems that Sting is part of the problem. So our study is actually saying, hang on a minute, we need to look at Sting in a different way, and we need to use therapeutics that target this drug in a different way so that we can do what we want. In this case, turn off the immune system to a very particular set of targets. Well, the particular connection we've made is we've connected Sting to a molecule called IDO, and IDO has been linked by us and others now to turning off the immune system. So that's the reason why in our particular case we found that link. That link wasn't known before. It was thought that Sting is an immune activating signal. And in fact, Sting, because it can induce IDO as well in the way we've defined it, can actually turn off the immune system as well. Well, we don't know all the answers yet. We've got to figure out what we call upstream and downstream parts of the pathway. So we're really still trying to figure out the exact pathway. The reason we do that in discovery science in the lab is to figure out yet more targets that can help us deliver a therapy in a much more focused way that reduces the amount of potential toxicities due to side effects of the drugs. And so the ultimate goal is to treat those diseases in a better way.